Welcome to this video in which we will see about development of microbiology as a discipline, discovery of microscope and microorganisms. The story of microbiology begins in the 17th century, a time when people could only guess that what caused diseases, decay or fermentation. The naked eye could see only the surface of life, but the invention of the microscope changed everything. In 1665, Robert Hooke, an English scientist, looked through the compound microscope at the thin slice of cork. To his amazement, he saw tiny compartments resembling honeycomb cells, which he called cells. Though Hooke did not actually see living organisms, his observation opened the door of microscopic world. His book Micrographia, filled with detailed sketches, inspired curiosity about the unseen. Meanwhile, in the Netherlands, a self-taught lens maker named Anton van Leeuwenhoek took the art of microscopy to a new level. Using simple microscope, he crafted himself with powerful lenses. He observed rainwater, scrapings from his teeth, and even paper infusion. What he saw astonished him. Tiny moving creatures that no one had seen before. He described them as animalcules. These were the first observations of bacteria, protozoan, and other microorganisms. Leeuwenhoek's letter to the Royal Society of London between 1673 to 1723 provided the world's first glimpse into the microscopic realm, making the true beginning of microbiology as a science. Abiogenesis versus biogenesis. Before microorganisms were discovered, people widely believed in abiogenesis, the idea that living things could spontaneously arise from non-living matter. This belief, supported by the philosopher Aristotle, claimed that worms came from meat, frog from mud, and flies from decaying matter. For centuries, no one questioned it seriously because there was no way to observe what truly happened on a microscopic level. In the 17th century, Italian physician Francisco Redi challenged this idea through the simple but elegant experiment. He placed piece of meat in 3 jars, one open, one covered with gauze, and one sealed. Maggots appeared only in the open jar where flies could reach the meat. Reddy concluded that living organisms' maggot came only from the other living organisms' flies, not from non-living material. Although his experiment weakened the idea of spontaneous generation, some scientists still believed that microorganisms could appear spontaneously. The final and decisive blow to abiogenesis came in the 19th century with the work of Louis Pasteur. Pasteur used specially designed swan neck flasks containing nutrient broth. The curved neck allowed air to enter but trapped dust particles. When the broth was boiled, it remained sterile unless the flask was tilted, allowing dust to reach the broth. Only then did microbial growth appear. This elegant experiment proved that microorganisms come from pre-existing life forms, not from non-living matter. Thus, Pasteur firmly established the theory of biogenesis. Supporting Pasteur's finding, the British scientist John Tyndall performed experiments demonstrating that dust carried microorganisms and that some bacterial spores could resist boiling. He introduced the concept of Tyndallization, a process of intermittent boiling to kill heat-resistant spores. With this, the controversy finally ended. The principle, life comes from life, became the cornerstone of modern biology and microbiology. Golden era of microbiology between 1857 to 1914. The late 19th and early 20th centuries are remembered as golden era of microbiology when brilliant scientists uncovered the microbial causes of many diseases and developed revolutionary methods for prevention and cure. Their discoveries not only advanced medicine but also transformed agriculture, food preservation, and our understanding of life itself. Louis Pasteur, the father of microbiology. Louis Pasteur, a French chemist, was one of the most influential figures in science. Initially studying fermentation, he discovered that microorganisms were responsible for the souring of wine and milk. This led him to invent the process of pasteurization, in which liquids are gently heated to kill harmful microbes while preserving the taste. Pasteur's work provided the first evidence that specific microorganisms causes specific biological change. Pasteur's most revolutionary idea was the germ theory of disease, which proposed that many diseases are caused by microbes, not by bad air or miasma, as previously thought. His discoveries led him to develop vaccines for several diseases, including anthrax in animals and rabies in humans. Pasteur worked on rabies, where he saved the life of a young boy bitten by a rabid dog, made him a hero. His research laid the foundation of immunology, preventive medicine, and sterile techniques in laboratories. Robert Koch, bacteriologist. Building on Pasteur's finding, the German physician Robert Koch developed techniques to identify and grow bacteria in pure culture. He discovered the bacteria that causes anthrax, Bacillus anthracis, tuberculosis, Mycobacterium tuberculosis, and cholera, Vibrio cholerae. To establish a direct relationship between a microbes and a disease, he proposed Koch postulates, a set of 4 principles that remains fundamental to medical microbiology today. Koch's contribution also includes the development of solid culture media using agar, staining techniques, and the use of microscope for diagnostics. His systematic approach transformed bacteriology from descriptive to an experimental science. For his discoveries, Koch was awarded the Nobel Prize in Physiology or Medicine in 1905. Together, Pasteur and Koch are considered the 2 pillars of medical microbiology. Joseph Lister, the pioneer of antiseptic surgery. During the same period, hospitals were dangerous places where infections claimed countless lives after surgery. Inspired by Pasteur's germ theory, Joseph Lister, a British surgeon, introduced the practice of antiseptic surgery. He used carbolic acid phenols to sterilize surgical instruments and clean wounds. As a result, the rate of post-surgical infections dropped dramatically. Lister's antiseptic techniques transformed surgical practice and made modern surgery possible. His methods also influenced the later development of antiseptic techniques, practice that prevent contamination in hospitals and laboratories. He is rightly remembered as the father of modern surgery. Paul Ehrlich, the founder of chemotherapy. Paul Ehrlich, a German physician, was fascinated by the interaction between chemicals and cells. He developed staining methods to differentiate various types of blood cells and bacteria, contributing to diagnostic microbiology. Ehrlich is best known for his concept of magic bullet, a chemical that could specifically target disease-causing microorganisms without harming the host. He discovered salvarsan, the first effective drug for the treatment of syphilis, making the beginning of chemotherapy, the use of chemicals to treat infectious diseases. Ehrlich also made pioneering contribution to immunology, including theories of antibody formation and concept of receptors on cell surfaces. Elie Machnikov, the discoverer of phagocytosis. Russian zoologist Elie Machnikov added another dimension to immunology. While studying starfish larvae, he observed certain cells engulfing foreign particles, a process he called phagocytosis. He proposed that similar cells in humans, called phagocytes, defend the body by swallowing and destroying the invading pathogens. Metchnikoff's discovery formed the basis of cellular immunology, complementing Pasteur's work on vaccines and early theories of antibodies. He later joined the Pasteur Institute where he continued his studies on aging and probiotics. For his contribution to immunology, Metchnikoff shared the Nobel Prize in 1908. Edward Jenner, the father of immunization. Decades before Pasteur, Edward Jenner, an English physician, made a life-saving discovery that laid the foundation of modern vaccination. He noticed that milkmaids who had contracted cowpox never suffered from smallpox, a deadly disease. In 1796, Jenner inoculated a young boy, James Phillips, with material from cowpox lesions and later exposed him to smallpox. The boy did not fall ill, proving that cowpox provided protection. This was the world's first vaccination. Jenner's discovery saved millions of lives and eventually led to the eradication of smallpox. His simple yet revolutionary idea demonstrated that diseases could be prevented through immunization, one of humanity's greatest medical discovery. Alexander Fleming, discovery of antibiotics. In 1928, Scottish bacteriologist Alexander Fleming made a chance discovery that changed medicine forever. While working with Staphylococcus bacteria, he noticed that mold Penicillium notatum had accidentally contaminated one of his petri dish and killed the surrounding bacteria. He identified the active substance as penicillin, the world's first antibiotic. Although large-scale production came later during World War II, Fleming's discovery marked the beginning of antibiotic era, saving countless lives from infections that were once fatal. Penicillin became the model for developing many other antibiotics, revolutionizing healthcare and surgery. Martin Beijerinck and Sergei Vinogradsky, pioneer of environmental microbiology. While most early microbiologists studied disease-causing microorganisms, Martin William Beijerinck and Sergei Vinogradsky explored the beneficial role of microbes in nature. Beijerinck, the Dutch scientist, developed the environment enrichment culture technique, allowing specific microorganisms to be isolated from mixed samples. He also discovered viruses as infectious agents smaller than bacteria while studying tobacco mosaic disease, coining the term virus. Russian microbiologist Sergei Vinogradsky focused on soil bacteria and discovered that their role in biogeochemical cycles. He identified bacteria capable of nitrification, sulfur oxidation, and nitrogen fixation, showing that microbes are essential for recycling elements in ecosystem. His work laid the foundation of microbial ecology and environmental microbiology, highlighting the importance of microbes beyond diseases. Conclusion: the journey of microbiology from Leeuwenhoek's animalcules to Pasteur's germ theory, Koch's postulates, and Fleming's antibiotics is a story of human curiosity and scientific progress. Each scientist built upon the discoveries of others, transforming microbiology from the curiosity into the one of the most powerful sciences that touch every aspect of life, medicine, agriculture, industry, and ecology. Together, they unveil the invisible world that sustains and protects all living beings. Thank you.